All right, let's jump right in. We're going to talk about something pretty incredible today, the massive digital archives that hold the secrets to life itself. We're talking about the world of biological databases. It's a story of unbelievable scale, of some really hidden challenges, and well, the quiet heroes who are keeping it all in check.
So just to give you an idea of the scale we're dealing with, look at this number, 54.2 petabytes. Let that sink in for a second. That is the size of just one of the major data archives, the European Nucleotide Archive. We're talking millions upon millions of gigabytes of raw biological code. So the question is, how on earth do we even begin to make sense of all of that?
Well, maybe the best way to think about it is like this. Imagine a single enormous library. And in this library is the blueprint for every single organism that has ever been studied. This isn't science fiction anymore. This is the reality of modern biology. So welcome to what we're going to call the Library of Life.
Now, the official term for this library is a biological database. It's basically a super specialized computer archive that's built to handle this absolute tidal wave of data. Everything from DNA sequences to the 3D shapes of proteins. And it organizes it so that scientists can actually find what they need and use it to make new discoveries.
And just like any good library, this one isn't just a chaotic pile of books. It has different sections. And for our purposes, the two most important sections are what you can think of as the original manuscripts and the curated encyclopedias. This distinction is absolutely key.
So here's how that all breaks down. On one side, you have your primary databases. These are the archives of that raw, original data submitted straight from the labs. This is the first-hand evidence. And once something goes in here, it's part of the permanent scientific record. Then on the other side, you have secondary databases. These are more like research guides or encyclopedias. They take that raw data, they analyze it, they organize it, and they present it in a way that helps us find new connections and discover new knowledge.
And to put some real names to these, your primary manuscripts for DNA sequences are held in these giants like GenBank. Protein structures, they live in the PDB. And then secondary databases like Uniprot and Ensemble come along, take all that primary data, and add these extra layers of analysis, like showing us how proteins are related or adding notes to entire genomes. They all have to work together in this huge, interconnected system.
Okay, but here's the twist. Here's where it gets complicated. You have millions of researchers submitting data for decades, using all sorts of different technologies and methods. What happens when the library starts to get, well, messy? What if the same book is on the shelf a dozen times, but with slightly different titles, or worse, with conflicting chapters inside?
And this brings us right to the heart of one of the biggest challenges in this whole field, what we're calling the duplicate dilemma. And believe me, it is a problem that goes way deeper than just having a few extra copies lying around.
So what do we actually mean when we say a record is a duplicate? It's not always an identical carbon copy. The best way to think about it is with this definition. Two records are duplicates if, for whatever job you're trying to do, having one of them means you don't need the other one. It's totally dependent on the context of your work.
And this is where that complexity really kicks in. A duplicate could be an exact sequence, sure, but it could also be a sequence that's just really similar. Or it might be a small fragment of another, longer sequence. It could even be a sequence that looks pretty different but actually represents the exact same biological thing. Untangling all of this is a surprisingly tricky problem.
And the stakes are really high. As the researchers who live and breathe this stuff say, this isn't just about wasting a little hard drive space. These duplicates, especially the ones that are inconsistent with each other, can actively mess up scientific analysis and lead you to the completely wrong answer.
Okay, let's make this real. We're going to look at a simple but super important measurement called GC content. It's just the percentage of two of the four DNA bases, guanine and cytosine, in a sequence. And scientists use this for everything from classifying a new species to designing basic lab experiments.
So, check out this chart. That first bar on the left, that shows a GC content calculation from a sample that's just full of messy, duplicate records. It comes out to 48.1%. But when that exact same sample is cleaned up, when the duplicates are properly dealt with, the real value, that middle bar, is actually 53.7%. That's a 5.6% difference.
Now you might be thinking 5.6%. So what? Well, in genetics, that is a massive gap. For certain microbes, a difference of just 3% is enough to tell two different species apart. So a data error like this, caused by nothing more than duplicates, is more than enough to send an entire research project down the completely wrong path.
Which brings us to the solution. If the library is messy, Who are these dedicated librarians working tirelessly behind the scenes to keep it all organized and accurate for everyone else?
They're called biocurators, and they are absolutely essential. It all starts when a researcher submits some new data. Then these expert curators and scientists have to manually review it, hunt down any potential duplicates, and merge them all into a single clean representative record that finally gets published. It is a painstaking, human-driven process of quality control.
So why on earth do we go to all this trouble? Why does having a clean, perfectly curated library of life actually matter out here in the real world?
Because this foundation is what everything else is built on. I mean everything. You want accurate medical tests? You need clean data. You want to discover life-saving drugs faster? You need clean data. You want to create better crops or truly understand evolution? Every single bit of it comes back to the quality and the integrity of this data. A clean library really does power the future of science.
And that leaves us with one final and really important question. We started this off with a number, 54 petabytes, but that number is growing exponentially year after year. So as this library expands to a size we can barely even imagine, the challenge of keeping it clean only gets bigger. Who will be the librarians of the future? And how will they make sure that the stories this incredible library has to tell us are actually true?